So for the fibromyalgia patients, here's the P2X4 receptor. This is control. This is the fibromyalgia patient. This is the TRPV1 receptor. This is control. This is for the fibromyalgia patient. This is the IL-10 gene, or cytokine gene. This is the fibromyalgia. This is the control. We found that 3 different genes here were highly statistically different in the fibromyalgia patient. They were higher, much higher than control. So that was a surprise to us. We really didn't anticipate that at all, that this was going to define the difference in fibromyalgia patients. And then we actually thought about it a little bit, and we recognized that this could be for several reasons. It might be part of the reason that they don't change that much following exercise. If they're already up, they got nowhere to go. Okay, so they do change some, they do a little worse, but it's not like the chronic fatigue patients that you saw before. It may be part of the reason that the American College of Pharmacology came up with that idea that they didn't really get worse following exercise. But they're already bad at baseline. It also could be that these people are, and this particular group of patients, because we were told to do this by our sponsors, were not allowed to be on any medications. And so it's possible what we got here was a group of patients that weren't that affected because they could go off all their medications or didn't have to take any medications in order to be in the study. And we lost the patients that were very, very affected. And that may be part of the reason that they have a different profile and don't show the gene increases following exercise. It's also possible that, because they're less affected, they're more active. So they're out walking around all the time, like most of you people aren't. So they're actually doing things, and as a result, they're turning their genes up during the day. And so they come in, when they come into the lab, their genes are already up, and therefore they do have less distance to go. They can't go up much more because they're already up, because they are more active to start with. We don't know the answer to that, but for whatever reason, this defines this group and not the chronic fatigue syndrome patients. Okay, so I just gave you all the answers here now. And you're going to, now there's so many questions you have, right? I'm going to go on and actually tell you more about that. It turns out that the gene expression that we look at follows the severity of the disease too. So these are patients with greater severity, these are patients with less severity. So these are the most severely affected patients, patients that are less severely affected, the less still affected, and the least affected patients. And you can see that the gene expression pattern matches that. And these are controls right here. So you can see that even down here, they're above the controls, but there is a sort of a match. So the gene expression does fit with how severe the symptoms are in these patients. Okay. Okay. But what are we really measuring? Gene expression, what is that? And why are we really measuring that? Okay, so this is the real part of the talk here. We'll come back to the other at the very end here. So what is gene expression? How does it differ from genetic causes? What causes change in gene expression? Why is gene expression changed in chronic fatigue syndrome and fibromyalgia syndrome? And how does it help chronic fatigue patients to know their gene expression is abnormal? So these are the real nuts and bolts questions, as opposed to just showing you data there. So first of all, genetics versus gene expression. Genetics is the result of the DNA coding you inherited from your parents. The genetic code of your DNA that you inherit is initially, at least, the same as in all the cells of your body. All your cells have the same DNA coding in it. The differences in the expression of the genes is why you have different tissues like heart, kidney, bones, and brain. So what makes the genes makes you different is not all the tissues in your body. What makes a bone as opposed to a brain is not the DNA that's there, because the DNA is all the same. It's the amount of expression of that DNA. How much of that DNA is actually made into protein, okay? So that's what makes it different. Okay, the cell nucleus contains the DNA you inherited from your parents, and this is a pretty picture of a cell that I ripped off the internet, and so I apologize to whoever I ripped off from. I should have put the attribution there. So this is the nucleus. This is where the DNA is. DNA is all the squiggly stuff I penciled in here. and this is penciled in DNA, so it's squiggly stuff, and it's really just really long strings, and there are 44 strings that you have tossed around in your nucleus because you have 44 chromosomes, and so each one of these little strings is broken up into a length, and so there are 44 lengths of these strings. They normally unravel all over the place. When the cell divides, they condense into really something you can see much better with a microscope. So normally it's all strung out all over the place. And all these little bends and loops and things, they mean something. Okay, the DNA is what makes up the chromosomes. It's usually stringy with many windings and meanderings around proteins that are located in the nucleus. So this is sort of the cartoon many of you have seen of DNA that's actually composed of a double helix with little cross bands like this that hold it together, and it's a double helix because it's both a helix to start with and it also bends around itself, and it can coalesce into chromosomes that have this sort of a pattern to it. But if you look at here, this is the code. These are the base pairs, C, A, and that one's T, I believe this is G. So there's only 3 of these, sorry, so it's got to be just a copy. There's only 3 on each of these sides, and they pair with base pairs on the other side here. So this is a T, G, C, A there. Okay, so this is the code. It's just that over and over again in a different order. All the way along this band here that actually defines exactly what kind of protein is going to be made. All right. So it is the code of life. The order of those base pairs is the code of the life. It is the specific order of that code that the bases are in that occur that actually is the code. And this is at A, T, C, G, T, A, T, and these can be in any kind of order, and except that they define, this order defines exactly what protein is going to be made by that DNA. All right. So this is the DNA, and there's those little cross bands. These are the base pairs right here. All right, so genetics is this code, or ordering of base pairs in the DNA. It's what defines all the proteins and more that make up your body, okay? So that winding I started showing you, the fact that DNA had little twists and turns in it, and was squiggly all over the place, the winding around the nucleoproteins and other special proteins called transcription factors, which are different in all tissue types. The winding is actually part of what defines what part of that DNA is actually going to be expressed, meaning going to be made into proteins. So if parts that are wound tightly around aren't going to be expressed, parts that are less tightly wound are going to be expressed, they're going to make proteins. And that's different in all the tissues in your body, and it's why a bone is a bone and your brain's a brain. So, okay. So in order for the DNA to make proteins that make up your body, it must next be transcribed into RNA. So that DNA must go through an intermediate step. It must be basically an RNA copy of that DNA must be made. And that's called transcription, DNA transcription. So the DNA can unzip itself. So the 2 sides of that thing come apart, and then those base pairs are exposed, and then an RNA copy is made on one of those sides along those base pairs. And the RNA copies a short segment of the DNA, and it makes a copy of that code. The RNA copy leaves the cell nucleus and directs the formation of a protein, and using this code, it copies. Using the code, it copied from the DNA. So this is the DNA, this is a cartoon, and the DNA unwinds a little bit here, forms a strand of RNA, and this is the mRNA. The RNA then goes out of the cell nucleus and then actually is bound to the ribosome, and the ribosome then actually attracts in, that's another transfer RNA. The transfer RNA actually has protein peptides on it. These peptides then, because they're bound to the transfer RNA, line themselves up and then form the polypeptide or the protein. So the protein is actually made from the RNA. Now, I go through all of this because this is what we measure. This mRNA, this messenger RNA, the RNA that will come out of the cell and actually start to make proteins. That's what all that gene expression that you saw is. We're measuring the amount of RNA for specific genes, for specific, that's going to make specific proteins. Okay, the DNA code is inherited, but the amount of RNA you made is affected both by the code, so the code affects the amount you make, and it's also affected by your environment. That's why I want to show you the difference between genetics and what we look at, which isn't genetics. It's actually gene expression. It's because the environment can affect it. So the cells can receive a signal. For example, the chemicals you make in your muscle when you're working. So the cells can get that signal. If they get that signal can activate a transcription factor, which is a protein. The protein can then go in and bind to the DNA. And in binding to the DNA, it can either cause that part of the DNA to be more transcribed, so you make more RNA, or less transcribed, so you make less RNA. You can have both positive and negative transcription factors. So and what that means is the chemicals that you're making can actually cause a change in transcription. So can a lot of other things. We'll go through some of the other things that can actually change the amount of DNA that's transcribed into RNA, which changes the amount of messenger RNA. So we're looking at the changes in that messenger RNA in our gene expression. So what we're looking at then is only partly what the DNA is made of, because if you've got a certain kind of DNA with a certain coding pattern, you may never be able to make as much RNA as somebody who has a different pattern. Okay, you start with that. You have that to begin with. However, the environment can change it even more than your inherited ability to make the RNA. The environment can change the amount of RNA you make even more than the pattern that's laid down by your inherited DNA. Okay. So if the protein being made is what signals the brain, in this example, that your muscle is tired, then you will sense more muscle fatigue, for example. And this is what we decided to measure. We decided to measure these particular types of RNAs as a biomarker for chronic fatigue syndrome and fibromyalgia. So what we decided to measure was the RNA that makes the proteins that signal muscle fatigue and muscle pain. That's why we picked those particular genes that we looked at to form the biomarkers. So this is the final part. It's the part that tells you that you're fatigued, that tells you that you have muscle pain. This is not the part that caused that signal that changed the RNA. So in fact, we're not looking at the causes. For example, virus might cause this. In fact, these are some of the things that can change it. Viruses can change the amount of RNA. Stress can change the amount of RNA. Pollutants can change the amount of RNA. Your diet can change the amount of RNA. Exercise can change the amount of RNA that you make. Lots of different things can affect the amount of RNA you make. Diseases of various varieties. Flu can change the amount of RNA you make. Viruses at that time as well. So why do we measure RNA as opposed to measuring the proteins? Well, this is sort of the lamppost version of this. RNA is easier to measure than the signaling proteins because it doesn't take much of these proteins to do the signaling. There's so little protein that it's really difficult to measure it. It's actually easier to measure RNA because we have really good tools to do that. And that's what this says basically. Rapid and accurate methods to measure RNA have been perfected in the last 10 years, and in fact in the last 3 or 4 years they've gotten really good. I don't know if any of you follow the cancer literature at all. There's an explosion going on there in terms of looking at gene expression because the tools are really available. It's really easy and much cheaper to do. Okay. So the RNA changes can give us clues as to what causes the body to increase fatigue signaling. The RNA changes can also give us clues to more permanent changes in the winding and regulation of the DNA that can occur with diseases and aging. So I was telling you about transcription factors. Well, they can temporarily change the amount of RNA, but in fact, there are ways to make those changes even more permanent. Some viruses will do that. Some other just consistent activation of transcription factors over time can actually change the regulation of DNA so that permanently you make more of a particular RNA, or permanently you make less, unless something very powerful happens to change that. So there are more permanent ways to do this as well.